everyone, I am Munquad. I am the research scholar of the Department of Biotechnology and IT Warangal, India. I am here to present my research work on the topic of the development of deep learning based diagnostic tools for glioma subtyping. Glioma is the type of a brain cancer, and high grade glioma, that is the glioblastoma multiforme, that is the GBM, is a highly invasive and devastating form of the primary brain cancer. So, GBM is frequently diagnosed in the older patient. The prognosis of the GBM is very poor. It has only 12 months median survival time. There is a need to develop a tool for diagnosis and select the appropriate drug for the GBM. The objective of my work is to develop the DL-based tool to classify the subtypes of GBM. Based on the histopathological method, GBM subtypes are classified into three classes, that is the classical, mesenchymal and proneural. Some challenges are there for the subtype classification of GBM, that is the molecular heterogeneity for the early diagnosis of the GBM, which leads to inaccurate therapeutic intervention. Importance of deep learning using transcriptome and epigenome data for cancer classification. It is reported that from the literature, transcriptome and epigenome biomarker have emerged as the promising tool for developing the precision medicine. Therefore, correct subtype prediction is important for therapeutic intervention, and genome-wide analysis reveals that alteration in the gene expression and methylation pattern, the GBM, are distributed throughout the genome. Therefore, the dimension of the oncogenic features is high, which cannot be dealt with a statistical approach. Therefore, DL method is essential as the oncogenic changes are distributed in different genomic layer and transcriptome and epigenome. So, multi-omics data integration is essential to classify the subtype to get the global view of the molecular signature of the disease, and recent advances in the deep learning algorithm also shows the efficiency in the data integration. Next, I have designed the deep learning based framework in the two approaches. In the first approach, transcriptome and methylome data are retrieved from the TCGA. In the pre-processing step, low expressed genes are removed. Next, I have selected the high and low methylated region of promoter, and up and down regulated gene that are linked with the promoter regions are selected based on the Z-score. Further, data is divided into the training and test data, and feature selection was done on the training data, and model machine learning based and deep learning based model were developed, and model evaluation was done by different parameters such as accuracy, recall, precision, MCC and G-mean. Next, model ranking was done using the multiple criteria decision making method, and external data was also used to validate the model. Separately, WGCNA is applied to understand the link between the co-expression of feature genes and GBM subtypes, and further to check the survival status of module genes by survival analysis. And next, I have applied the PCA to check the distribution of GBM subtypes. PCA was done on the pre-processed data and feature selected from lasso, and we can see that in the pre-processed data, PCA is failed to separate the GBM subtype, and lasso features are able to distinguish the GBM subtypes. Next, I have applied different machine learning and deep learning methods to classify the GBM subtypes, and we can see that CNN, that is the convolutional neural network, performs better among all other machine learning algorithms in the training data and validation data set. It achieved greater than 90% accuracy, but only from one parameter we cannot say our model is good. So I have selected several more parameters, that is recall, precision, F1, FPR, G-mean and MCC. After that, I have applied multiple criteria decision making method to rank the machine learning and deep learning algorithm, and CNN got the first rank among other machine learning algorithms. It is always desirable to have a highly sensitive and specific model for diagnosis. So I visualize the relationship between the sensitivity and specificity using the ROC plot for the CNN model, and it achieved the AUC score of 0.91%. Next, WGCNA is applied to understand the link between the expression of the feature genes and GBM subtypes, and different color modules are formed, and each module having a set of genes that different modules are positively and negatively associated with the GBM subtype shows by the heat map, and each set of modules that are enriched in the cancer associated pathways and genes to check the survival status of the module gene by survival analysis. I found that subtype specific biomarkers are identified in the each subtype like CCDC in the classical, mesenchymal RAF38, and in the proneural TOLIP. These could be a drug target for the subtype of GBM. In the second approach, transcriptome and methylome data are taken from the TCGA and high and low methylated regions of promoter and up and down regulated genes that are linked with the promoter region are selected based on the Z score in the subtype specific manner. Next, cog regression was applied to select the cancer-associated genes and methylated regions, and these cancer-associated genes and methylated regions are separately fetched into the autoencoder that generates the latent variables for the creation of deep learning based model, and model evaluation was done by the external data set. This is the basic architecture of the autoencoder, in which encoding layer, bottleneck layer and decoding layer is present. In the encoding layer, input data, that is the gene expression and methylation data, are compressed and generates new latent variable in the bottleneck layer by concatenating the gene expression and methylation sites, and in the decoding layer, that is a mirror image of encoding layer from the bottleneck layer, that latent variables were taken for developing a deep learning based model by ANN and CNN classifier, and model evaluation done by the external. Next, the model performance of ANN and CNN, we can see that in the GBM data, like in the training data, it got the accuracy of greater than 90%, and the validation data also it got the accuracy of greater than 90%. Next, I compare the deep autoglioma to the other methods, such as model developed by the random features in the pre-processed data. Deep autoglioma performs outstanding than the other method. Next, geoanalysis was done on the cancer-associated genes and methylated regions that shows the cancer-associated pathways and processes are enriched. Approach 2 is more biologically informed because cancer-associated genes and methylated regions are selected before data integration and model development. In approach 1, lasso features are selected regardless whether it is cancer-associated or not. Approach 1 shows the higher accuracy, but approach 2 is more biologically informed and avoids black box problem and favors the clinical diagnostic. These are the references, and I acknowledge the NIT Warangal and matrices for the fellowship.